Okay. I hope my slides are visible and I'm audible. Yeah. So good evening, everyone. Today is the 12th live session of this lecture series. Basically, in these live sessions, as you know, we discuss some of the questions, previous year assignment questions from the course Biochemistry, taken by Professor K. Subramaniam. And in these live sessions, we basically discuss some of the previous year assignment questions. And we not only discuss the questions, we try to discuss the concepts behind those questions so that it's easier for us to solve similar type of other questions, right? So, yeah. So, today we'll be discussing some problems and we'll try to discuss the concept behind those problems. So I hope my slides are visible and I am audible. If at any point I become inaudible or my slides are not visible anymore, please let me know. Okay. So we'll move on to the first question for today. The second half of glycolysis is also known as the payoff phase because the ATP spent in the first half of glycolysis is recovered in this phase. The total number of ATP molecules generated in this payoff phase and the net gain or loss in the number of ATP molecules over the whole of glycolysis are dashed and dashed respectively. Correct? Which one of these options do you think is correct? Anybody? Anybody? We'll discuss the questions once you let me know what you think about it.
I'm not sure.
Okay, so we'll discuss the answers. So the answer will be 4 and 2, and why we'll discuss glycolysis and basically the investment phase and the payoff phase in detail. So we know what are the objectives of the process of glycolysis. It's a major oxidative pathway for glucose. The main reactions of the glycolytic pathway is a rate limiting enzymes so regulation which basically regulate the process. ATP production can be aerobic or anaerobic, as we know. It can be fermentation, which will be anaerobic, or aerobic if we continue with the further processes. ATP production is thus both can be aerobic and anaerobic, and pyruvate kinase deficiency will maybe lead to hemolytic anemia. So, if we take an overview of glycolysis, it's a major pathway for glucose oxidation. It occurs in the cytosol of all cells. It is unique because in that it can function either aerobically or anaerobically depending on the availability of oxygen and intact mitochondria. It allows tissues to survive in presence or absence of oxygen, example in skeletal muscle. And RBCs which lack the mitochondria are completely reliant on glucose as a metabolic fuel and metabolize it by anaerobic glycolysis. Correct? So if you check these mechanisms, this is aerobic glycolysis, this is anaerobic glycolysis. So this is basically the process of glycolysis which ultimately leads to the Krebs cycle. But if you are focusing on only glycolysis, this is glycolysis, and this is how this is the aerobic glycolysis, this is anaerobic glycolysis. Okay, so most of it is same. The only difference being that after pyruvate, it will go on towards Krebs cycle and oxidative phosphorylation, whereas for anaerobic glycolysis, it will probably go towards the fermentation steps. So in aerobic glycolysis, this glucose gets converted to glucose 6-phosphate with hexokinase and glucokinase. Hexokinase is present in most tissues, while glucokinase is present in some of the hepatocytes. Okay. Then there is a regulational process step, basically where fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. Okay, and this is caused by phosphofructokinase 1, and ATP is converted to ADP. So ATP and citrate basically negatively regulate this process, whereas AMP and fructose 2,6-bisphosphate, they basically positively regulate this process. This will give rise to fructose 1,6-bisphosphate. Okay, and then fructose 1,6-bisphosphate will finally give rise to glyceraldehyde 3-phosphate or dihydroxyacetone phosphate, and these two can be with the help of aldolase, and these two can interchange amongst each other with the help of triose phosphate isomerase. So phosphofructokinase 1 is a regulationary step. Then they talked about the aldolase and triose isomerase, role of aldolase and triose isomerase. Now, when we move on with the glyceraldehyde 3-phosphate, it gets converted to 1,3-bisphosphoglycerate. The NAD plus is taken in and is converted to NADH plus H plus, and with the help of this enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, so for each NADH, 3 ATP will be produced by the electron transport chain in the mitochondria. That is 6 ATP will be produced. That is the furthest, that comes under the further steps. Then from 1,3-bisphosphoglycerate, the action of mutase can give rise to 2,3-bisphosphoglycerate, which can also get converted to 3-phosphoglycerate, or 1,3-bisphosphoglycerate can directly get converted to 3-phosphoglycerate using phosphoglycerate kinase. And 2,3-bisphosphoglycerate can get converted to 3-phosphoglycerate because one phosphate from the 2 position needs to be removed, so phosphatase. Okay, now 3-phosphoglycerate will get converted to 2-phosphoglycerate by using phosphoglycerate mutase. Okay, so you can see all the steps and all the enzymes that are carrying out these steps, and then 2-phosphoglycerate will give rise to phosphoenol pyruvate, PEP, phosphoenol pyruvate, with the help of the enzyme enolase and with the release of water molecule. Then PEP, PEP or phosphoenol pyruvate will give rise to pyruvate with the involvement of pyruvate kinase. So this process is positively regulated by fructose 1,6-bisphosphate. So ADP is converted to ATP. Now, phosphoglycerate kinase, the step of phosphoglycerate kinase where ADP gets converted to ATP is known as one level of substrate level phosphorylation. And the other step where pyruvate kinase, initiates pyruvate kinase basically is involved in the process of conversion of phosphoenol pyruvate PEP to pyruvate. That process also known as substrate level phosphorylation. In both this process, ADP is converted to ATP. Now, what is the difference between substrate level phosphorylation versus oxidative phosphorylation? So, phosphorylation, if you know, is the basic metabolic reaction of introducing a phosphate group into an organic molecule. Now, for oxidative phosphorylation, the formation of high energy phosphate bonds by phosphorylation of ADP to ATP coupled to the transfer of electrons from reduced coenzymes to molecular oxygen via the electron transport chain, it occurs in the mitochondria. And for substrate level phosphorylation, the formation of high energy phosphate bonds by phosphorylation of ADP to ATP, or it can be guanosine diphosphate to guanosine diphosphate, GTP to GTP, coupled to cleavage of a high energy metabolic intermediate, which is the substrate. So that is the difference between oxidative phosphorylation and substrate level. Substrate level phosphorylation can also happen in cytosol. Okay. So see, these two levels are phosphate substrate level phosphorylation. Okay. Now, in this case, there's a covalent modification that can happen in pyruvate kinase. So, if you know about the basic cell signaling pathways, we know that there is a receptor on the cell surface which can bind to glucagon. The binding of the receptor will activate this adenylate cyclase, which will convert ADP to CAMP plus PPI, inorganic phosphate and cyclic adenosine monophosphate. So this will help in the activation of protein kinase A. Now this protein kinase A, what it does is that it adds a phosphate group to pyruvate kinase. Pyruvate kinase in itself is active. But when this protein kinase A adds a phosphate to pyruvate kinase, it becomes inactive. Okay, so now PEP will get converted to pyruvate. This process. This is a covalent modification of pyruvate kinase, which may have a role in the regulation of the glycolysis because it is this pyruvate kinase enzyme is involved in the conversion of PEP to phosphoenol pyruvate to pyruvate. Now, what can be the long-term regulation of glycolysis? Insulin. Insulin will induce the process of glycolysis, and glucagon will repress the process of glycolysis. So, at each of these steps, insulin and glucagon can have different opposite contradictory effects on the synthesis of key enzymes of glycolysis in liver. This is a summary of regulation of glycolysis. The regulatory enzymes are the irreversible reactions like glucokinase or hexokinase, phosphoryl 1 and pyruvate kinase. Regulatory mechanisms can be rapid, short-term like allosteric or covalent modifications. It can be slow, long-term like induction and repression. So, that is how regulation of glycolysis takes place. Okay. Now, what happens when there is a pyruvate kinase deficiency? It can cause hemolytic anemia. Okay, so here if you see the enzyme may show an abnormal Km or Vmax. If you have known, we have also discussed about Michaelis-Menten equations in one of the previous classes. So the enzyme may show an abnormal Km or Vmax for substrates or coenzymes, and this enzyme may show an abnormal response to the activator fructose 1,6-bisphosphate. So PK or pyruvate kinase mutation can give rise to altered enzyme kinetics, decreased enzyme stability, and altered response to activator. So these are the cons of for pyruvate kinase, because a mutation can lead to all of these. And we talk about aerobic glycolysis and ATP production. So the ATP consumed in the entire process will be 2. ATP produced into at substrate level phosphorylation, it's 2 ATP produced. But there are 2 steps. So 2 into 2, 4. For oxidative level, there are 3 steps, but 2 ATP produced each step, so 6 ATPs. The total will be 10 ATPs. But we know that 2 ATPs were also consumed. Which step consumed the ATPs? Anybody? So this step consumed ATPs. If you see, ATP is consumed and ATP is formed. Right? This step also consumes ATP. So 2 ATPs were consumed. So that's why the net ATP production will be 8, because 10 minus 2 is 8. So the total versus net ATP production. So in the first few steps are known as the energy investment phase, whereas the next few stages are known as the energy generation phase. So for aerobic glycolysis, these are steps: glucose to 2 pyruvate, 2 ADP to 2 ATP, 2 NAD plus to 2 NADH. Now for anaerobic glycolysis, what happens? These processes are same, but pyruvate gets converted to lactate, and NADH is consumed in this process to NAD plus. These 2 steps are substrate level phosphorylation. So this is the role of lactate dehydrogenase. What happens after pyruvate? In other cases, it will go towards the Krebs cycle, but in case of anaerobic conditions, there will be fermentation basically. So the lactate dehydrogenase will act on the pyruvate to convert it to lactate with the production of NAD plus. So what is the net ATP production in case of anaerobic glycolysis? So the ATP consumed will be 2, which is same. ATP produced will be via substrate level phosphorylation 4, and oxidative level will be 6. Okay. Sorry, so oxidative level will not be there, right? So only substrate level phosphorylation will be there, which is 4. This will not be there because this is anaerobic glycolysis. Okay, so the total ATP produced is 4. ATP consumed will be 2. So 4 minus 2 is 2 ATP. So you see, for anaerobic glycolysis, the difference is so evident, because only 2 ATP net production is happening, whereas in aerobic glycolysis there are 8 ATP net production is happening. See, yeah. So anaerobic glycolysis can have an RBCs 2,3 BPG shunt. So this is all about anaerobic glycolysis and aerobic glycolysis that we have already talked about. So in case of glycolysis in RBCs, these are the steps of ATP production, and these are the probable number of net ATPs generated in RBCs. So in RBCs, the end product will be lactate, no net production or consumption of NADH. NGL will be not 2,3 BPG or 2,3 BPG shunt, 2 ATP and 0 ATP, and the hemolytic anemia will depend on all these factors. So if you're talking about glycolysis and RBCs, this will be important. So what is the 2,3 bisphosphoglycerate shunt basically? So it's basically a metabolic pathway in mature erythrocytes or RBCs involving the formation of 2,3 bisphosphoglycerate or 2,3 BPG which regulates oxygen release from hemoglobin and delivery into tissues. Okay, so this 2,3-BPG, the reaction product of the Lubbolding and Rappaport pathway, was apparently first described and isolated by an Austrian biochemist. Okay, so throughout the Lubbolding-Rappaport pathway, bisphosphoglycerate mutase catalyzes the transfer of a phosphoryl group from C1 to C2 of 1,3-BPG, giving rise to 2,3-BPG. 2,3-bisphosphoglycerate, the most concentrated organophosphate in the erythrocyte, forms 3-phosphoglycerate by the action of bisphosphoglycerate phosphatase. The concentration of 2,3-BPG varies proportionally with the pH, since it is inhibitory to catalytic action of bisphosphoglyceromutase. Okay. Now, glycolysis is a major oxidative pathway for glucose. Glycolysis is employed by all tissues. It is a tight rate regulated pathway. PFK1 is a rate limiting regulatory enzyme, okay, because it has a role in regulation. So, glycolysis is mainly a catabolic pathway for ATP production, but it has some anabolic features as well. And pyruvate kinase deficiency in RBCs results in hemolytic anemia. So, for aerobic glycolysis, the net production of ATP is 8. For anaerobic glycolysis, the net production of ATP is 2. For glycolysis in RBCs without 2,3 bisphosphoglycerate synthesis, it is 2 ATP, but with 2,3 bisphosphoglycerate synthesis, it is 0 ATP. So this is the glycolytic pathway. The reactions of glycolysis, we can all find that in this book. White and White book is very famous biochemistry. You can check out this book as well. This is just the pathway shown in extreme detail. This is the glycolysis we've already talked about all the different. So after pyruvate is formed, either we can go for anaerobic homolactic fermentation where lactate is generated, or we can go for anaerobic alcoholic fermentation where alcohol is generated, ethanol is generated, and aerobic oxidation where it goes to a citric acid cycle and then it goes to an oxidative phosphorylation. So we know that glucose is converted to pyruvate while generating 2 ATPs. 2 molecules of NAD plus are converted to 2 molecules of NADH. So the oxidizing power of NAD plus must be recycled. So pathway overview, overall reaction is this: glucose to pyruvate, NAD plus to NADH, ADP to ATP plus PI and to then H2. If we do the pathway overview, there are 10 enzyme catalyzed reactions considered to occur in 2 stages. Stage 1 is the preparatory phase, where glucose is phosphorylated and cleaved to yield 2 molecules of glyceraldehyde 3-phosphate. Stage 1 uses 2 ATPs, whereas stage 2, overall reaction, the payoff phase, where 2 gaps are converted to pyruvate and generation of 4 ATPs. So see, the stage 1 is the preparatory stage where glucose is phosphorylated and cleaved to yield 2 molecules of glyceraldehyde 3-phosphate. It is gaps. Stage 1 uses 2 ATPs and stage 2 generation of 4 ATPs. This is the glycolytic pathway. You can see this, exactly what we have discussed. I'll leave it here. You can check the pathway out for 1 minute. Okay, so next the reactions of glycolysis. So stage 1 preparatory phase we have already talked about. Hexokinase, the first ATP utilization, phosphoglucose isomerase, phosphofructokinase 1, second ATP utilization, aldolase, triphosphate isomerase. These are the first stage 1 preparatory phase reactions of glycolysis. You can see all the reaction schematics over here only. Then the preparatory phase. This is hexokinase, role of hexokinase with Mg2 plus. Then in reaction 1 of glycolysis, the transfer of phospharyl group from ATP to glucose to form glucose 6 phosphate. Now hexokinase and its complex role with glucose purple molecule over here. Okay, so the enzyme substrate complex, the two lobes gray and green over here swing together to engulf the substrate. This excludes H2 from the active site, which prevents ATP hydrolysis. Step 2 is phosphoglucose isomerase or phosphohexose isomerase. So phosphoglucose isomerase catalyzes the conversion of G6P to F6P. G6P was glucose 6 phosphate and F6P is fructose 6 phosphate. And the conversion of an aldose to a ketose. Now the isomerization of an aldose to a ketose. So the open chain representations over here show the existence of the reaction. Now if you talk about step 3 phosphoglucokinase, it phosphorylates fructose 6 phosphate in reaction 3 of glycolysis, second ATP utilization. So phosphoglucokinase plays a central role in control of glycolysis because it catalyzes one of the pathways rate limiting determining reactions. So this is second ATP utilization for phosphoglucokinase to fructose 6 phosphate to fructose 1,6 bisphosphate. Step 4 is aldolase. Aldolase catalyzes the cleavage of fructose 1,6 bisphosphate in reaction 4 of glycolysis. So this forms two trioses, glyceraldehyde 3-phosphate gap or dihydroxyacetone phosphate, which is DHAP. Give me a second. Step for aldolase, aldol cleavage of fructose bisphosphate to form two trioses, glyceraldehyde GAP, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. I can check this terminology out. Next step, triphosphate isomerase. So only gap continues along the glycolytic pathway. Dihydroxyacetone phosphate is the ketose and glyceraldehyde is the 3-phosphate and aldose. Okay. Now the fate of the carbon atoms of glucose in the formation of glyceraldehyde 3-phosphate. So in case of dihydroxyacetone, these all are derived from the glucose carbon. This part might be in the formation of glyceraldehyde 3-phosphate. Since stage 2 pair phase, glyceraldehyde 3-phosphate dehydrogenase is first high energy intermediate formed. Phosphoglycerate kinase, first ATP generation, phosphoglycerate mutase, and enolase second high energy intermediate formation, and pyruvate kinase will be second ATP generation. These are the one sentence descriptions of these all enzymes. And step 6, glycerol phosphate dehydrogenase is the first high energy intermediate formation. So you can see glycerol phosphate dehydrogenase, and there is 1,3-phosphoglycerate at the bottom of the slide, which is being converted from glyceraldehyde 3-phosphate. So this is the reaction of glyceraldehyde 3-phosphate dehydrogenase. Now, what are the enzymatic mechanisms of GAPDH? It forms an enzyme-substrate complex first, then thiohemiacetal intermediate, then a little bit of acyl thioester intermediate, and then NAD plus and NAD plus. All of these are being produced. Now, phosphoglycerate kinase is also helping in the first ATP generation. This is phosphoglycerate kinase and Mg2 plus. Now, phosphoglycerate kinase produces ATP generation. So, upon substrate binding, the two water domains of PGK and phosphoglycerate kinase are two domains of PGK swing together to permit substrates to react in a water-free environment, as occurs with hexokinase. This is a mechanism of PGK reaction. So, what are the genetics of the overall GAPDH PGK? It's written here. Step 8 is the phosphoglycerate mutase wala, where 3-phosphoglycerate gets transformed to 2-phosphoglycerate. Takes away 1-phosphate group. What is the reaction mechanism of phosphoglycerate mutase or PGM? Catalytic amounts of 2,3-phosphoglycerate are required for the enzymatic activity. Inclusion of the enzyme with catalytic amounts of labeled 2,3-VPG is a 32-phosphate labeled enzyme. So what is the reaction mechanism of PGM? Go check out the slide. This is self-explanatory. From 2-carbon phosphoryl enzyme complex, everything starts happening. Cyclic, like not cyclic exactly, it's a sequential process. Now, glycolysis induces influence of oxygen transport. So, 2,3-VPG binds to the deoxyribonucleic hemoglobin and alters the oxygen affinity of hemoglobin. The erythrocytes synthesize and degrade 2,3-VPG by a detour from the glycolytic pathway. A lower BPG in erythrocytes as resulting from hexokinase deficiency results in increased hemoglobin oxygen affinity. Next is step 9 enolase, where this is the second high energy intermediate formation by dehydration mechanism. So pyruvate kinase is the second mode of living generation. However, tautomization of enol pyruvate to pyruvate keeps on happening. So that was all regarding the aerobic and the anaerobic oxidation process. If you have any doubt, you can ask me, or we can move on to the next question. The next question is something like this. The conversion of the phosphoglycerate to 2-phosphoglycerate is catalyzed by the enzyme dash, and the active site of this enzyme consists of single amino acid which mediates the entire reaction. A. Phosphoglycerate isomerase aspartame. 1,3-phosphoglycerate mutase histidine, phosphoglycerate mutase histidine, and bisulfite 3-phosphate isomerase histidine. So you have to do the match and pair. Anybody? Take a few minutes and think about it. No hurry. Anybody? You can think about it in 2 minutes and then we'll discuss. We just discussed about which of the enzymes 3-phosphoglycerate to 2-phosphoglycerate. What is the mechanism? Which enzyme will be involved?
The third one.
Okay, phosphoglycerate mutase.
Yeah, like this.
Okay, so we'll check the glycolysis pathway only. 1 second. Yeah, so the answer is phosphoglycerate mutase, and the amino acid responsible that mediates the entire reaction will be histidine. So again, this is the pathway basically. So if you see from 3-phosphoglycerate to 2-phosphoglycerate, this is the enzyme, phosphoglycerate mutase. Okay, we have talked about the front end of glycolysis in details. So if we talked about the different enzymes, phosphoglycerate kinase will be the first ATP generation step. This is the reaction of phosphoglycerate mutase. So 3-phosphoglycerate, 2-phosphoglycerate, 2,3-phosphoglycerate. Now, phosphoglycerate mutase requires the phosphorylated form of the enzyme to be active. Only 2,3-bisphosphoglycerate can phosphorylate the unphosphorylated enzyme. That somehow confers some kind of specificity to the process. See, there are so many histidine amino acids in the active site. the phosphohistidine residue. So you see how this happens. It's a 2 phosphoenzyme complex. Okay, give rise to phosphoenzyme, then 3 PG phosphoenzyme complex, then 2,3-BPG enzyme complex, and then ultimately 2-diphosphoenzyme, inactive diphosphoenzyme, and histidine is on its own. Now, glycolysis apparently also influences oxygen transport. Okay, but if you look at this reaction mechanism, glycerylate 3-phosphate gap DH to 3 phosphoglycerate, and then phosphoglycerate mutase from 3-phosphoglycerate to 2-phosphoglycerate. So enolase helps in the generation of a second high energy intermediate, like from 2-phosphoglycerate to phosphoenol pyruvate. This is how from 2-phosphoglycerate phosphoenol pyruvate is made by a carbon ion intermediate. And pyruvate kinase will be the second intergeneration step. Phosphoenol pyruvate plus ADP will give rise to ADP, and enol pyruvate will ultimately give rise to pyruvate. Second half of glycolysis is the payoff phase. So we know the generation of high energy compound, substrate level phosphorylation, rearrangement, generation of high energy compound, and substrate level phosphorylation again. So what is the metabolic fate of pyruvate? Either alcoholic fermentation, ethanol and CO2, or homolactic fermentation, lactate, a citric acid cycle to CO2 and H2O. So what is the need to generate NAD plus from NADH? So homolactic fermentation is a conversion of pyruvate to lactate, and mammals have 2 different types of enzymes, isozymes. Okay, I will for now use this rotation. M will be used for muscles and H type will be for heart. So lactate dehydrogenase is a tetramer. H4 has a low Km affinity for pyruvate and is allosterically inhibited by high concentrations of pyruvate. Whereas muscle isozyme has a higher Km for pyruvate and is not allosterically regulated. So all the 5 types can exist: H4, H3M, H2M2, HM3M4. The M or the muscle one predominates in anaerobic muscle tissues which favor the formation of lactate, while the H1 or the normal one form predominates in aerobic tissues like heart, where the formation of pyruvate from lactate is preferred. Now, if you talk about alcoholic fermentation, it's basically a 2-step process. A pyruvate decarboxylase requires thiamine pyrophosphate TPP as a cofactor. Alcohol dehydrogenase requires Zn2 plus as a cofactor. If you talk about thiamine pyrophosphate, so this buildup of negative charges seen in the carboxylation reactions on the carbonyl atom in the transition state is unstable, and thiamine pyrophosphate helps stabilize the negative charge. So what is the reaction mechanism of pyruvate decarboxylation? So nucleophilic attack by the yield from form of TPP on the carbonyl, departure of CO2 and resonance stabilization of the carbon ion, then protonation of the carbon ion and elimination of TPP yield to form acetaldehyde. So what does the deficiency of thiamine pyrophosphate can cause? It can lead to beriberi. So beriberi is prevalent in the rice consuming countries of the Orient where polished rice is preferred. So DPP is found in the brown outer layers of rice. Deficiency can cause neurological atrophy, cardiac failure, edema, nowadays found in alcoholics who would rather drink than eat. So glycolysis is about 100 times faster than oxidative phosphorylation in the mitochondria. Okay. So what is the meaning of fast twitch muscles? The short bursts of energy and are nearly devoid of mitochondria used exclusively glycolysis for ATP. The slow twitch muscles are dark red, rich in mitose obtained from ATP from oxphos, and fried muscles of migratory bats and the muscles of long distance runners. We've also talked about how any kind of protein phosphorylation may activate or inactivate a protein. So, what are the three steps to elucidate common controlling mechanism pathway? Identifying the rate determining steps, those with the large negative delta G, and measure flux to the pathway, and each step with inhibitors. How we can identify in vitro allosteric modifiers for the pathway, study each enzyme with enzyme kinetics, mechanisms, and inhibition patterns. Or we can measure in vivo levels of modulators under conditions consistent with the proposed control mechanism. There are only three enzymes that can function with large negative delta Gs: hexokinase, phosphofructokinase, and pyruvate kinase. Other enzymes operate in equilibrium and their rates are faster than the flux through the pathway. So ATP concentrations only vary about 10% from resting to active cells. ATP is buffered by creatine phosphate and adenylate kinase. So 10% decrease in ATP produces a fourfold increase in AMP or adenosine monophosphate because ADP 1 ATP will be 50 adenosine monophosphate in muscles. So AMP activates BFK by the action of adenylate kinase. What is substrate recycling? Fructose 6 phosphate to fructose 1 6 bisphosphate. Fructose 1 6 bisphosphate to fructose 6 phosphate. The net result is a breakdown of ATP. The two different enzymes control this pathway, PFK and fructose 1 6 bisphosphate. If these both were not controlled by a futile cycle, would occur. We talked about the entire mechanism. We will not go into very much details. These are the very much details we can discuss about it once we are done with the other questions. Moving on to the next question. In the various alternate feeder pathways of glycolysis, the intermediates of glycolysis are obtained from other metabolic pathways and proceed to glycolysis. Which of the following substrates and the corresponding pathways act as feeder pathways to glycolysis? These are the four options. Take a minute or 2-3 minutes and then we can start discussing this question. Okay, so answer will be these are the feeder pathways, alternate feeder pathways of glycolysis from mannose to mannose 6 phosphate, from mannose 6 phosphate to glucose 6 phosphate. The other will be sucrose to fructose, fructose to fructose 1 phosphate to glyceraldehyde, dihydroxyacetone phosphate, and glyceraldehyde 3 phosphate. Anyone will like comment on this, or we'll discuss more about it? These are the feeder pathways for glycolysis. And so this is trehalose here. This is glucose. Lactose can come via lactase to glucose. Trehalose can also come. Sucrose can also come as sucrose to fructose. Fructose will directly come to glucose 6 phosphate. These are the basically the various ways with the feeder pathways for glycolysis works. Okay, so feeder pathway of galactose is also like this. Galactose, if excessive accumulation happens, it can give rise to galactonic acid or galactositol. Galactositol, galactose will be converted by galactokinase to galactose 1 phosphate, which will get converted from galactose 1 phosphate to glucose 1 phosphate via galactose 1 phosphate uridyl transferase. Okay, so this is, and galactose 1 phosphate to glucose 1 phosphate is easy. Now glucose 1 phosphate can be converted to UDP glucose via UTP PPI. UTP will be converted to PPI, and UDP glucose pyrophosphorylase. UTP galactose 4 epimerase is also involved in the process. So these are the feeder pathways. Trehalose can get converted to glucose using trehalase. Okay, so these are the feeder pathways of glucose. So if we come back to the question that we started with, mannose can be converted to mannose 6 phosphate to finally glucose 6 phosphate. And sucrose can be converted to fructose, then fructose 1 phosphate, glyceraldehyde plus dihydroxyacetone phosphate, and then glyceraldehyde 3 phosphate. Okay. So we'll move on to the next question for today. Gluconeogenesis and glycolysis are essentially reverse processes, but some enzymes of gluconeogenesis take part in glycolysis as well, hence having reversible activity depending on which substrate it acts on. Which of the following enzymes catalyze steps of both glycolysis and gluconeogenesis? Phosphoglycerate mutase, phosphofructokinase 1, phosphoexose isomerase, pyruvate kinase, phosphoglycerate kinase. So, anybody? Anybody would like to try? Anybody would like to try? Anybody would like to try?
I created 500 something.
Okay. 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 So gluconeogenesis glycolysis, obviously the name suggests, uh, gluconeogenesis is the new genesis of something related to glucose. And glycolysis is obviously breakdown of glucose. So obviously these are essentially reversible processes. Okay. But obviously some enzymes of one process can take part in the other process. That means that enzyme can have reversible activity. Depending on the substrate, it will show that kind of activity. Okay. So basically phosphoglycerate mutase, phosphohexose isomerase and phosphoglycerate kinase. These three will basically have this kind of reversible activity depending on the substrate it acts on. So glycogen metabolism. This is the structure of glycogen. Glycogen is a homopolysaccharide formed of branched alpha D glucose units. So the main glycosidic bond is alpha 1,4 linkage. Only at the branching point the chain is attached by alpha 1,6 linkage. So each branch is made up of 12 to 14 glucose units. Okay, and glycogen is present mainly in cytosol of liver and muscles. Correct. So this is, if we talk about glycogen metabolism, for liver glycogen, the source will be blood glucose and other kind of hexose like fructose and the non-carbohydrate. For muscle glycogen, it will be blood glucose. So the amount will be 120 gram maximally depending on the liver weight. Muscle glycogen will be 400 gram maximally. Concentration will be 6% in liver glycogen, 1% in muscle glycogen. Another function. So source for blood glucose for the body, especially the brain at times of need. After 12 to 18 hours of fasting, liver glycogen is depleted. Whereas muscle glycogen, the source of energy for contracting muscles and producing lactate, is that. So what is the end product of glycogenolysis? Glucose for liver glycogen. Sorry. And lactate for muscle glycogen. And if you talk about hormonal regulation, so adrenaline, noradrenaline, and glucagon stimulate glycogenolysis and inhibit glycogenesis, and insulin has an opposite effect. Whereas in case of muscle glycogen, adrenaline and noradrenaline stimulates glycogenolysis and inhibits glycogenesis, and insulin has an opposite effect. So glucagon has no effect essentially. So what is glycogen? It's a deformation of glycogen in liver and muscle. Glycogen metabolism, glycogenesis genesis is synthesis of glycogen basically. So formation of glycogen in liver and muscles. So what are the substrates for glycogen synthesis? Blood glucose in liver, other hexose like fructose and galactose, and non-carbohydrate sources gluconeogenesis like glycerol and lactase. So these are converted first to glucose, then to glycogen. In muscles, it is blood glucose only. Now, for glycogen metabolism and synthesis of glycogen, glucose 6 phosphate will be converted to glucose 1 phosphate and then to PPI to PI. And from UDP glucose pyrophosphorylase, UDP glucose will be converted to UTP, and then glycogen synthase will act on it. So there is a tyrosine chain, which is the glycogenin, and glycogen synthase will act on it, and the branching enzyme will be 4,6 transferase. So further elongation at the non-reducing ends where glycogen synthase will be making alpha 1,4 bonds. Further branching will be making the alpha 1,6 bonds. So that is how glycogen synthesis happens basically. Now glycogen metabolism and synthesis of glycogen steps. So glucose molecules are the first activated to uridine diphosphate glucose. Then these UDP glucose molecules are added to a glycogen primer to form glycogen. Now formation of UDP glucose. Okay, so formation of glycogen. So UDP glucose reacts with glycogen primer, which may be few molecules of glucose linked together by alpha 1,4 linkage, and a protein called glycogenin. So UDPG molecules react with OH of tyrosine of that protein to initiate glycogen synthesis. Okay. And then glycogen synthase enzyme. So by action of the glycogen synthase with the key enzyme of glycogenesis, UDPG molecules are added to glycogen primer, causing elongation of the alpha 1,4 branches up to 12 to 14 glucose units. Correct? And the branching enzyme will be transfers parts of the elongated chains 5 to 8 glucose residues to the next chain, forming a new alpha 1,6 glycosidic bond. So the new branches are elongated by the glycogen synthase and the process is again repeated. So if we talk about glycogen metabolism and breakdown of glycogenolysis, it's the breakdown of glycogen into glucose in liver and lactic acid in muscles. Just if we phosphorylase, the key enzyme of glycogenolysis acts on alpha 1,4 bonds, breaking it down by phosphorylasis. It is breaking down by addition of inorganic phosphate. Therefore, it removes glucose units in the form of glucose 1-phosphate. Now, phosphorylase enzyme will act on the branches containing more than 4 glucosyl units. So, when the branch contains 4 glucose units, 3 of them are transferred to the next branch by transferase enzyme, leaving the last one. So, the last glucose unit that is attached to the original branch by alpha 1,6 bond is removed by debranching enzyme by hydrolysis. That is breaking the bond down by addition of H2. Then glucose 1-phosphate molecules are converted to glucose 6-phosphate by the mutase enzyme. Now, what is the fate of glucose 6-phosphate? In liver, glucose 6-phosphate is converted to glucose by glucose 6-phosphatase. In muscles, there is no glucose 6-phosphatase, so glucose 6-phosphate enters glycolysis to give lactate. Now, if we talk about glycogen metabolism regulation, there is a coordinated regulation of glycogenesis and glycogenolysis. That is the conditions leading to stimulation of glycogenolysis, inhibiting at the same time glycogenesis and vice versa. So what happens during fasting when you are starving? Glycogenolysis is stimulated and glycogenesis is inhibited. Obviously, because the body needs something right now. So this provides blood glucose from liver glycogen. So after meal, part of the absorbed glucose goes to general circulation to be utilized. So the remaining 60% is converted into glycogen in liver. So after meal, glycogenesis is stimulated and glycogenolysis is inhibited. So the principal enzymes controlling glycogen metabolism are glycogen synthase and phosphorylase. Now what happens during fasting? During fasting, the binding of hormones such as glucagon or epinephrine to membrane receptors signals the need for glycogen to be degraded, either to elevate blood glucose levels or to provide energy for exercising muscle. So this leads to activation of the protein kinase. So binding of glucagon or epinephrine to the specific cell membrane receptors results in the cyclic adenosine monophosphate mediated activation of CAMP dependent protein kinase. So this enzyme is a tetramer having 2 regulatory subunits and 2 catalytic subunits. So cyclic adenosine monophosphate binds to the regulatory subunit dimer, releasing individual catalytic subunits that are active. So then glycogen metabolism and regulation. So activation of phosphorylase kinase. So phosphorylase kinase exists in 2 forms, an inactive B form and an active A form. So active cyclic AMP dependent protein kinase phosphorylates the inactive B form of phosphorylase kinase, producing the active A form. Activation of the glycogen phosphorylase. So glycogen phosphorylase also exists in 2 forms, the dephosphorylated inactive B form and the phosphorylated active A form. So active phosphorylase kinase phosphorylates glycogen phosphorylase B to its active A form, which then begins glycogen breakdown. So phosphorylase A is reconverted to phosphorylase B by the hydrolysis of its phosphate by protein phosphatase 1. So if you see, this is glucagon, liver and muscle will be for muscle and over here. So what is the role of calcium in muscle? During muscle contraction, calcium is released from sarcoplasmic reticulum. So calcium binds to the calmodulin subunit of phosphorylase kinase, activating it without phosphorylation. So phosphorylase kinase can then activate glycogen phosphorylase, causing glycogen degradation. Correct? So what is the role of AMP, adenosine monophosphate, in muscle? So in muscle, under extreme conditions of anoxia and depletion of ADP, AMP activates glycogen phosphorylase B without it being phosphorylated. So if you can see over here, glycogen is finally degraded. Once the glycogen phosphorylase B is converted to glycogen phosphorylase A, inactive to active form, the glycogen is degraded. So inactivation of glycogen synthase. So the regulated enzyme in glycogen synthase is glycogen synthase. So it also exists in two forms, the active A form and the inactive B form. However, for glycogen synthase, the active form is dephosphorylated, if you see over here, whereas the inactive form is phosphorylated. So glycogen synthase A is converted to the B form and therefore inactivated by phosphorylation at several sites on the enzyme with a level of inactivation proportional to its degree of phosphorylation. This conversion process is catalyzed by several different protein kinases that are regulated by cycling AMP or other signaling mechanisms. So the binding of the hormones glucagon or epinephrine to the hepatocyte receptors or of epinephrine to muscle cell receptors results in the activation of adenylyl cyclase mediated by a G protein. So this enzyme catalyzes the synthesis of cyclic AMP, which activates cyclic AMP dependent protein kinase A as described. Okay, so protein kinase A then phosphorylates and thereby inactivates glycogen synthase. Glycogen synthase B can be transformed back to the A form by protein phosphatase 1, which removes the phosphate groups hydrolytically. So glycogen metabolism after meal. So blood glucose level tends to increase, stimulating the release of insulin hormone. So insulin stimulates the phosphodiesterase enzyme, converting cyclic AMP into AMP, abolishing its glycogen degrading effect. Now, insulin also stimulates phosphatase enzyme that removes phosphate group from glycogen synthase enzyme. Is activating it, and phosphorylase enzyme is inhibiting it. So as a consequence, the glycogenolysis decreases and glycogenesis is induced. Okay, so if you see this cyclic AMP, so from ATP adenylyl cyclase will convert to cyclic AMP, then cyclic nucleotide phosphodiesterase will convert it to AMP. Now, what is glycogen metabolism and glycogen storage diseases? So, these are a group of inherited disorders characterized by deposition of abnormal type or quantity of glycogen in the tissues. So, they are mainly due to deficiency of one or many enzymes of glycogen metabolism. Okay. So, type 1 can be von Gerl's disease. So, it can be caused due to the deficiency of glucose 6 phosphatase and is most common type. It is characterized by accumulation of large amount of glycogen in liver, leading to disturbance of liver functions and hepatomegaly. The symptoms can be fasting hypoglycemia, ketosis and hyperlipidemia, hyperuricemia, that is basically gout. So decreased glucose 6 phosphate, increased glucose 6 phosphate, decreased glucose 6 phosphatase will lead to increase in glucose 6 phosphate, stimulating pentose phosphate pathway, increasing ribose production, increasing uric acid, then basically leading to gout. Type 2 can be Pompe's disease. Type 3 can be limit dextrosis or Cory's disease. Due to deficiency of deep branching enzymes in liver, muscles, and heart, glycogen has many short branches, as we know. Now, if we talk about gluconeogenesis, it is the formation of glucose from non-carbohydrate sources. These sources typically include lactate, pyruvate, glycerol, some amino acids like glucogenic amino acids, and propionate. So, what are the functions? Gluconeogenesis supplies the body with glucose. So, glucose is the only source of energy for nervous tissues, RBCs, and skeletal muscles during exercises. So, glucose is the precursor of milk sugar and lactose in mammary gland. And glucose is important during low carbohydrate diet or when liver glycogen is depleted. Okay, it is depleted after 12 to 18 hours. So, gluconeogenesis clears the blood from the waste products of other tissues as lactate. produced by muscles and RBCs. So location will be intracellular, location will be cytosol and mitochondria. Organ location will be liver or kidney. Now for gluconeogenesis, major pathways and regulation of gluconeogenesis and glycolysis in the liver. So entry points of glucogenic amino acids after transamination are indicated by the arrows extended from these circles, if you can see. And the key gluconeogenic enzymes are shown in the double bordered boxes. And the ATP required for gluconeogenesis is supplied by the oxidation of fatty acids, and propionate is only important in ruminants. If you see the arrows like this, signify allosteric effects, and dash shafted arrows like this over in modification by the reversible phosphorylation. And high concentrations of alanine can act as a gluconeogenic signal by inhibiting glycolysis at the pyruvate kinase step. See over here, alanine is acting as a gluconeogenic signal. So the steps of gluconeogenesis are mainly the reversal of glycolysis, as we said, except for the three irreversible kinases which are replaced by the following enzymes like pyruvate and phosphoenol pyruvate. So reversal of the reaction catalyzed by pyruvate kinase in glycolysis involves two endothermic reactions. Okay, mitochondrial pyruvate carboxylase catalyzes the carboxylation of pyruvate to oxaloacetate, an ATP requiring reaction in which the vitamin biotin is the coenzyme. Biotin then binds to CO2 from bicarbonate as carboxybiotin prior to the addition of the CO2 to pyruvate. So the resultant oxaloacetate is reduced to malate, exported from the mitochondrion into the cytosol, and then oxidized back to oxaloacetate. And a second enzyme, such as phosphoenol pyruvate carboxykinase, catalyzes the decarboxylation and phosphorylation of oxaloacetate to phosphoenol pyruvate using GTP as the phosphate donor. So in liver and kidney, the reaction of succinate thiokinase in the citric acid cycle produces GTP rather than ATP as in other tissues. And this GTP is used for the reaction of phosphoenol pyruvate carboxykinase. Thus, it provides basically a link between citric acid cycle activity and gluconeogenesis to prevent excessive removal of oxaloacetate for gluconeogenesis, which would impair citric acid cycle activity. Now, fructose 1,6-bisphosphate and fructose 6-bisphosphate. The conversion of fructose 1,6-bisphosphate to fructose 6-phosphate for the reversal of glycolysis is catalyzed by fructose 1,6-bisphosphatase. So, its presence determines whether a tissue is capable of synthesizing glucose or glycogen not only from pyruvate but also from triose phosphates. So glucose 6-phosphate and glucose. The conversion of glucose 6-phosphate to glucose is catalyzed by glucose 6-phosphatase. So it is present in liver and kidney, but absent from muscle, which therefore cannot export glucose into the bloodstream. So blood glucose is derived from the diet, gluconeogenesis and glycogenolysis. So the digestible dietary carbohydrates yield glucose, galactose and fructose that are transported to the liver by the hepatic portal vein. Now, galactose and fructose are readily converted to glucose in the liver, and glucose is formed from two groups of compounds that undergo gluconeogenesis. So those that involve direct net conversion to glucose, including most amino acids and propionate, and those that are products of the metabolism of glucose in tissues. So lactate formed by glycolysis is in skeletal muscles and erythrocytes. It is transported to the liver and kidney, where it reforms glucose, which again becomes available by the circulation of oxidation in the tissues. So this process is known as Codai cycle or lactic acid cycle. Now in the fasting state, there is a considerable output of alanine from skeletal muscle, far in excess of the amount in the muscle proteins that are being catabolized. So it is formed by transamination of pyruvate produced by glycolysis of muscle glycogen, and it is exported to the liver, where after transamination back to pyruvate, it is a substrate for gluconeogenesis, right? So this glucose alanine cycle provides an indirect way of utilizing muscle glycogen to maintain blood glucose in the fasting state. So the ATP required for the hepatic synthesis of glucose from pyruvate is formed by the oxidation of fatty acids. Glucose is also formed from liver glycogen by glycogenolysis. So blood glucose is derived from the diet, gluconeogenesis and glycogenolysis. So what are the hormonal regulation of gluconeogenesis? So glucocorticoids like cortisol stimulates gluconeogenesis by the following mechanisms. They induce the synthesis of gluconeogenesis enzymes, and glucocorticoids stimulate protein catabolism by tissues. These two increase glucogenic amino acids available for gluconeogenesis. Then glucagon, it stimulates gluconeogenesis by lowering the level of fructose 2,6-bisphosphate. Insulin inhibits gluconeogenesis. It acts as an inhibitor for synthesis of enzymes of the gluconeogenesis pathway. And for the allosteric effects, there is acetyl-CoA and ATP. It stimulates gluconeogenesis by inhibiting glycolysis through inhibiting phosphofructokinase 1 and stimulating gluconeogenesis by stimulating the fructose 1,6-bisphosphatase. Acetyl-CoA also stimulates pyruvate carboxylase by the means of gluconeogenesis, and it inhibits pyruvate dehydrogenase by the means of oxidation. So it's an endogenic process. Gluconeogenesis is for the conversion of two molecules of pyruvate into one molecule of glucose, four molecules of ATP, two molecules of GTP, and two molecules of NADH plus H plus. So this is the 2 pyruvate to 2 oxaloacetate, 2 ATP is consumed, 2 oxaloacetate to 2 phosphoryl pyruvate or PEP, 2 GTP is consumed, 2,3 phosphoglycerate to 2,1,3 bisphosphoglycerate, 2 ATP is consumed. And for NADH plus H plus, 2,1,3 bisphosphoglycerate to 2 glyceryl di3-phosphate, 2 NADH plus H plus is consumed. Now, if we talk about glycolysis, it's the body's most readily available source of energy, glucose. And after digestive process, it breaks polysaccharides down into monosaccharides, right? Including glucose. So the monosaccharides are transported across the wall of the small intestine and the circulatory system, which ultimately transports them to the liver. Now, in the liver, hepatocytes then either pass the glucose on through the circulatory system or stores excess glucose as glycogen. So cells in the body can take up the circulating glucose in response to insulin, and through a series of reactions called glycolysis, they can transfer some of the energy in glucose to ADP to form ATP. So the last step of glycolysis produces the product pyruvate. So glucose plus 2 ATP plus 2 NAD plus 2 4 ADP plus 2 P plus P raised to pyruvate plus 4 ATP plus 2 N. So one glucose molecule will break down into two pyruvate molecules and creates two net ATP molecules and two NADH molecules by glycolysis. So therefore, glycolysis generates energy for the cell and creates pyruvate molecules that can be processed further through the aerobic Krebs cycle or the TCA cycle or citric acid cycle. It can be converted into lactic acid or alcohol in case of fermentation. or it can be used later for the synthesis of glucose to gluconeogenesis. So, as I was saying, glycolysis includes the enzymes responsible for the reactions. So, when glucose enters the cell, the enzyme hexokinase or glucokinase in the liver rapidly adds a phosphate to convert it to glucose 6 phosphate. Now, a kinase is a type of enzyme that adds a phosphate molecule to the substrate. So, in this case, glucose, but it can be true of other molecules also. Now, this conversion step requires 1 ATP and essentially traps the glucose in the cell, preventing it from passing back through the plasma membrane, thus allowing glycolysis to proceed. It also functions to maintain a concentration gradient with higher glucose levels in the blood than in the tissues. Now, by establishing this concentration gradient, the glucose in the blood will be able to flow from an area of high concentration to an area of low concentration to either used or stored. Now, hexokinase is found in nearly every tissue in the body. So, glucokinase on the other hand is expressed in tissues that are active when blood glucose levels are high, such as the liver. So, hexokinase has higher affinity for glucose than glucokinase and therefore is able to convert glucose at a faster rate than glucokinase. This is important when levels of glucose are very low in the body, as it allows glucose to travel preferentially to those tissues that require it more. So now the enzyme glucose 6-phosphate isomerase converts glucose 6-phosphate into fructose 6-phosphate. And like glucose, fructose is also a 6-carbon containing sugar. The enzyme phosphofructokinase 1 then adds one more phosphate to convert fructose 6-phosphate into fructose 1,6-bisphosphate, another 6-carbon sugar using another ATP molecule. Aldolase then breaks down this fructose 1,6-bisphosphate into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay. Now the triose phosphate isomerase enzyme then converts dihydroxyacetone phosphate into second glyceraldehyde 3-phosphate molecule. Therefore, by the end of this chemical priming or energy consuming phase, one glucose molecule is broken down into two glyceraldehyde 3-phosphate molecules. So the second phase of glycolysis, the energy yielding phase, creates the energy that is a product of glycolysis. So glyceraldehyde 3-phosphate dehydrogenase converts each 3-carbon glyceraldehyde 3-phosphate produced during the energy consuming phase into 1,3-bisphosphoglycerate. So this reaction releases an electron that is then picked up by NAD plus to create an NADH molecule. NADH is a high energy molecule like ATP, but unlike ATP, it is not used as an energy currency by the cell. Because there are two glyceraldehyde 3-phosphate molecules, two NADH molecules are synthesized during this step. Now each 1,3-bisphosphoglycerate is subsequently dephosphorylated. That is, the phosphate is removed by phosphoglycerate kinase into 3-phosphoglycerate. And each phosphate released in this reaction can convert one molecule of ADP into one high energy ATP molecule, resulting in a gain of two ATP molecules. The enzyme phosphoglycerate mutates and converts the 3-phosphoglycerate molecules into 2-phosphoglycerate. And the enolase enzyme then acts upon the two phosphoglycerate molecules to convert them into phosphanol pyruvate molecules. Okay, so the last step of glycolysis involves the dephosphorylation of the two phosphoenol pyruvate molecules by pyruvate kinase to create two pyruvate molecules and two ATP molecules. So this is the entire step by step of glycolysis as we had already discussed. So for anaerobic respiration, we know that when oxygen is limited or absent, pyruvate can enter a cell using anaerobic pathway, and in these reactions, pyruvate can be converted into lactic acid. In addition to generating an additional ATP, this pathway then serves to keep the pyruvate concentration low. So glycolysis continues, and it oxidizes NADH into NADH plus NAD plus. Needed by glycolysis. In this reaction, lactic acid will replace oxygen as anaerobic respiration, and this is an effective pathway of ATP production for short periods of time ranging from seconds to a few minutes. So the lactic acid produced diffuses into the plasma and is carried to the liver, where it is converted back into pyruvate or glucose via the Cori cycle. Similarly, when a person exercises, muscles use ATP faster than oxygen can be delivered to them. So they depend on glycolysis and lactic acid production for rapid ATP production. Okay. For aerobic respiration, we know that in the presence of oxygen, pyruvate can enter the Krebs cycle, where additional energy is extracted as electrons are transferred from the pyruvate to the receptors NAD plus TDP and FAD, with the carbon dioxide being a waste product. So the NADH and FADH2 pass electrons onto the electron transport chain, which uses the transfer energy to produce ATP. So as the terminal step in the electron transport chain, oxygen is the terminal electron acceptor and creates water inside the mitochondria. So as the terminal step in the electron transport chain, oxygen is the terminal electron accepted and creates water inside the mitochondria. So these are the basic steps of what happens. So at first glycolysis happens, then Krebs cycle, and then the ETC will have, electron transport chain will happen. So the differences between aerobic and anaerobic processes. We have talked about the gluconeogenesis, how the synthesis of new glucose molecules from pyruvate, lactate, glycerol, and this process exists primarily in the liver during periods of low glucose, that is under conditions of fasting, starvation and low carbohydrate diets. So why would the body create something fair amount of effort to break down certain key organs, including the brain, can use only glucose as an energy source. Therefore, it is essential that the body maintain a minimum blood glucose concentration. So when the blood glucose concentration falls below that certain point, new glucose is synthesized by the liver to raise the blood concentration to normal. So gluconeogenesis is not only simply the reverse of glycolysis, there are some important differences. Pyruvate is a common starting material for gluconeogenesis. So first the pyruvate is converted into oxaloacetate. Oxaloacetate then serves as substrate for the enzyme phosphoenol pyruvate carboxykinase, which transforms oxaloacetate into phosphoenol pyruvate. From this step, gluconeogenesis is nearly the reverse of glycolysis. So phosphoenol pyruvate is converted back into 2-phosphoglycerate, which is converted to 3-phosphoglycerate, and then 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate and then into glyceraldehyde 3-phosphate. The two molecules of glyceraldehyde 3-phosphate then combine to form fructose 1,6-bisphosphate, which is then converted into fructose 6-phosphate and then into glucose 6-phosphate. So we have talked about all the steps of glycolysis. This is a repetition of the same things. So basically, if you talk about both the processes, the different enzymes, so the different enzymes involved in glycolysis act as kinases, mutases, and dehydrogenases, cleaving enzymes, isomerases, or enolases. So they act in concert to split or rearrange the intermediates, so to add on phosphate groups and to move those phosphate groups onto ADP to make ATP. Several of the reactions involve the phosphorylation of intermediates, which is important not only for the production of ATP from ADP, but also as a useful handle on the substrate for enzyme binding to trap intermediates within the cell and to drive pathways in one direction by making phosphorylation and dephosphorylation reactions irreversible. So different enzymes have been split into two groups, those in phase 1 and those in phase 2. So for phase 1 will be obviously hexokinase, phosphoglucose isomerase, phosphofructokinase, fructose bisphosphate aldolase, all of these. So fructose bisphosphate aldolase catalyzes the reversible reaction of fructose 1,6 bisphosphate to 2 trans phosphates, both of which can continue through glycolysis, as you have seen. Okay. So there are different enzymes that have different catalytic functions, like aldolases A and C are mainly involved in glycolysis, while aldolase B is involved in both glycolysis and gluconeogenesis. So that is what we are talking about. So two key enzymes that basically regulate irreversible steps in these two processes, glucogenesis and glycolysis, are pyruvate kinase and phosphenol pyruvate carboxykinase, which catalyzes the last and the first step of glycolysis and gluconeogenesis respectively, and are both apparently regulated by lysine acetylation. Okay. So that was all. That is the question that we started with today. So we discussed both glycolysis and gluconeogenesis. So we will stop here today. If you have any questions, you can try to discuss, or we will stop here today. Anybody? Any questions? Okay, so there's no question. We can conclude the meeting today and stop the session. Okay, thank you.